of how these things work. First, I just want to draw a little bit of the sequence. Just and then we'll have some general things about these specific transcription factors, and we'll give a few examples. Okay, here's the promoter again. I'm going to represent as P. We'll have exon 1, E1, and intron, and exon 2. Okay. Okay. So that's the beginnings of the gene. Now, we have the promoter. Here's our big basal complex. Now, there are numerous sequences. Sometimes the common term for them is the term enhancers. Let's put that one down here. Okay, an enhancer is a common term for a DNA sequence that's going to bind some one or more specific transcription factors. Now, there are other names for them. And the term enhancer first came out when they first discovered, they found that this sequence, when present, could up the transcription of a given gene by 10, 100, even 1,000-fold. So they said it enhanced or increased transcription. Later on, they found some sequences seemed to have the opposite effect. It actually inhibited transcription. And they sometimes called them silencers. So silencers are DNA sequences that repress transcription. Now, the term silencer isn't used all that often except for, man from uncle type assassination. Okay. The reason why is they find out that many of these enhancers, many times the same enhancer, you bind one protein to it, transcription goes up, you bind something else to it, or regulate that protein, and transcription goes down. So there really aren't any too many, what you say, pure enhancers or pure silencers around. Most of these sequences could either increase or decrease transcription depending what's bound to it. Okay, so those are the ones that seem to repress transcription, but now we know that most of them can do both. Okay, now another common term, you'll see this in the literature quite a bit, because they tend to be located in front of the gene, not always where you see. They sometimes call them upstream regulatory elements, or UREs. You see that widely. Okay. Whatever you call these things, the key point is this. These are DNA sequences that bind specific transcription factors and either increase or decrease the transcription of that gene. And each gene has numerous, sometimes a score or more, separate enhancers scattered around the area of the gene. So what we can do here, we can put a number of enhancers. Some of them are located, I'll put E for enhancers, some of them are located quite close to the gene. By quite close, we're talking a few tens to a few hundreds of base pairs away from the promoter. Some enhancer sequences overlap with others. That's possible. That means you're going to have competition between what binds there. If something binds to this one, it may prevent something binding to the overlapping partner. Now, that's kind of like what we saw in the case of the promoter and the operator in prokaryotes. The sequences overlapped. So one protein binding to the operator could actually physically prevent the polymerase from binding to the promoter. So we can have overlapping enhancers as well. And we'll say tens, hundreds of base pairs. Okay. Now, you sometimes also have enhancers that are located several hundred to a couple of thousand base pairs away from the promoter. So we have enhancers that are often very far away. way, thousands of base pairs in some cases. Now there's a certain limit, but people have done interesting, when they first discovered these sequences, people were using recombinant DNA techniques to basically do things like move enhancers around, and they found some interesting things. They didn't know how they worked back then, but they found some interesting things about enhancers. You could take an enhancer and move it around quite a bit of a distance, and it would still work. There was a certain point, if you moved it far enough away from the gene, it wouldn't work anymore. But sometimes that far away meant several thousand base pairs. So you could flip you take these, and you take this one and move it over here, or this one and move it way out over here, and often they still work. So that says something kind of interesting. That raises the question, how does a DNA sequence that binds a protein have an effect on something that's 2,000 base pairs away? That's really pretty far away. When they have like internet connections, call up the big, text the, yeah, they got smartphones. Text the basal complex and say, hey, I'm here, fall apart, get out of here. Okay, there's actually another mechanism. But somehow, sometimes these guys are going to have to affect things, a huge promoter that can be a couple thousand base pairs away. They also found other things. You can flip these things around. You can reverse them. Like so. Splice it back in, and lo and behold, it still works. So they're orientation independent. You can flip them upside down, you can flip them backwards, and they'll still work. Sometimes, it's not too common, but sometimes they find enhancers located not in front of the gene. Occasionally you find some behind the genes. That's not too common, but once in a while you see something like that. People have found enhancers located inside introns within the gene. So these things can operate in a wide range of different locations. Now here again, the majority of genes, most or all of the stuff's going to be in front of the gene, not behind it, not in an intron sequence or something like that. But that can and does happen on occasions. Okay, let's get rid of the dot here. All right. Here, there, okay, so it says something about how these proteins must work. They are, seems to be, you can flip the sequence around, they still bind whatever proteins are supposed to. They can somehow work good distances, long distances away from the promoter. Yet we said before, somehow they interact with the basal complex. That's the key. All of them work somehow by affecting the basal complex, stabilizing it or disrupting it. So, brings up several points here, how these things work. Now, when I say the word specific transcription factor, so I don't have to write all that out, why don't I just put SPF, specific transcription factor here, okay? I'll spell that one time. In many cases, what we're going to see is that transcription factor will bind to the enhancer, and then the other end of it goes and binds to some part of the basal complex. So in other words, a direct interaction between the transcription factor and the basal complex. And that can either hold things down or push things off and destabilize. Now when you do that, of course, because proteins are fairly small molecules, they're large by molecule size, but they're small by our standards, there's going to be a limit. If you're going to directly bind the basal complex, it probably means that your enhancer has to be fairly close to the basal complex so you can reach over and grab it. Okay, so many of the ones that are fairly close to promoter can do that. So we'll say must be close to promoter. Okay, and the second thing that can happen is not so much that the transcription factor directly binds to the basal complex. Instead, it binds to an adapter protein or series of adapter proteins. And these adapter proteins then bind to the basal complex. And many of these adapter proteins are very large proteins, molecular weights of hundreds of thousands. In the case of one thing that we'll mention later, it's a complex of a whole bunch of them that has a molecular weight in the millions. So huge, gigantic. Something that big can reach over a long distance. So we could say indirect So by going through adapter proteins, you can effectively increase your risk, or your reach, and interact with the basal complex, even though you're quite a distance away. So that's another thing we can see. So we have both direct binding and indirect binding or regulation. And we'll see a couple mechanisms of that in just a little bit. Okay.
Does it bind to the basal complex?
No, we'll bind to a protein. Or a complex of proteins on one end, and the other end, I'm going to describe this a little bit later too,
Okay.
and the other end of that complex of proteins will bind to the basal complex. And it may stabilize things, it may push things off.
Okay.
So picture like an adapter. Case in point, if you go out to Europe, they use 220-volt lines. If you're going to run your 120-volt equipment, you have to get an adapter. You plug the adapter into the wall outlet, and then you plug your 120-volt thing into the adapter. And so the adapters a go-between between the two of them. Now, if you just plugged it directly in, of course, you'd burn out whatever, fry out whatever you had, and it wouldn't work too well. So you have to have these adapters. In this case, we use a lot of adapter proteins to do this. And it also has a pretty cool aspects of that too. We'll see that a little bit later. Okay, so we have the capability of direct binding and indirect binding. Now I'd like to mention several things about these specific transcription factors enhancers. Some key points here. Okay, first thing. A specific transcription factor may bind just to one enhancer exclusively and not any others. But it could also bind to multiple enhancers, to several different enhancers. And that means that that transcription factor may regulate a whole bunch of different genes. So, one or more enhancers. Some specific transcription factors are extremely cell-specific. For instance, the one that binds to enhancer in front of the globin genes, that one is found only in the red blood cell precursors, not in other cell types. So that's very specific. Only one of the several hundred different types of cells in our body has that particular transcription factor. And it binds the globin enhancer and fires off huge levels of transcription in them. So we have some that are very specific. You have others that are quite broad range. They are found in numerous different cells. And they activate transcription of many different genes in a whole wide range of cell types. A good example of that is a protein called MYC, M-Y-C. It turns out the problem with that is when that protein is improperly regulated or produced where it's not supposed to be, that's a well-known what they call oncogene. In other words, many cancers, you find problems with that particular one. Because the problem is, when that's around, it activates transcription of a wide range of genes and a wide range of cell types. Cell types that aren't including genes that are involved in regulating cell division. If you happen to have that in a cell you don't want, it can stimulate cell division that you don't want. So you have some that are very broad-based and others that are very specific. Okay? So they can either be very specific, they can also be fairly broad-based, where you find them in numerous different cell types and they regulate numerous different genes. So that's a second important one. Okay, a third point. A given enhancer. Now there are some enhancers that will bind. Question?
Yeah, what is that last thing underlined on number 2?
Oh, okay. Compete with.
No, number 2.
Oh, number 2. Okay, broad base. All right. I had fun handwriting in elementary school consistently. They throw up their hands in utter despair. Okay, once again, going through this. Specific transcription factors can be very specific to a single cell type, for instance. Or they can be very broad-based. Multiple cell types have those transcription factors, and they regulate transcription of numerous different genes. Okay, next thing. Each enhancer, some enhancers will bind one transcription factor, and that's it. But others can bind more than one different type of transcription factor. So what's going to happen in a case like this is if an enhancer can bind 3 or 4 or 5 different transcription factors, they're going to end up competing with each other. They're going to have to duke it out, and only one of them is going to bind at any given point. And what that means is the concentrations of the ones that can bind are very important. Concentrations are important. If you have one cell type, let's suppose you got 2 cell types, they both have the same enhancer, and each one of them can bind the same 3 transcription factors. Cell type 1, our transcription factor A is in abundance and B and C are not. So most of the time A is going to bind, and let's say that turns a gene on. Cell type 2, maybe there's equal amounts of all 3 of the transcription factors, so it's kind of a crapshoot as to which binds. And another cell type, you may have high levels of B, and that's more likely to bind than the others. And maybe that shuts the gene off. So you have these kind of things where you're going to have competition for the same enhancer in many cases. And whatever happens to that gene is determined on the concentration of the different transcription factors that can bind. They're all competing for the same binding sites, the same parts of the same enhancer. So that can add additional complications. And also you can have cell with a certain enhancer, certain transcription factor at one point in development, and maybe that turns the gene on, and then later on you start producing a different transcription factor that can also bind to the same enhancer, yet it has the opposite effect, it turns the gene off. So you can say, oh, during this period of development we're switching this gene on, and after it differentiates a certain point we'll just switch this gene off again. Just by varying levels of certain transcription factors. So that's a neat little thing. Another thing, a given enhancer, you may find this particular enhancer is in front of, or wherever, in front of one gene only. It will regulate that gene and nothing else. But there are some enhancers where you find the same enhancer sequences in front of many different kinds of genes. So if you have the appropriate transcription factor or factors, what's going to happen in this case is many genes can be regulated by the same protein. And that gives us the ability for one single protein to switch on or off a whole battery of different genes. Now I'll give you a good example. Steroid hormones. Steroid hormones bind to a protein called the steroid receptor. Each steroid has its own receptor or receptors. The combination of the hormone and receptor is a transcription factor. And there are many genes that have these so-called steroid response elements. They're enhancers that bind these steroids and the steroid and the receptors. So a single thing can have diverse effects in different cell types. Well, example for you ladies, almost the entire class here, right? Okay. About once every month, yeah, again, it's getting hard to use examples where the Y chromosome set because there's almost none of us here. Okay, that's an Asian-like trend, by the way. But if you look at steroid hormones, let's suppose that monthly estrogen spike. Okay, it's going to bind to and regulate gene expression in numerous cell types. The oocytes are going to be stimulated to undergo their final maturation, the completion of meiosis 1, ovulation. The cells in the uterine lining are stimulated to proliferate a great deal. Certain cells, certain neurons in your central nervous system are going to be stimulated to maybe overproduce neurotransmitter receptors and stuff so that those neural pathways are a lot touchier, a lot more active than they used to be. So now the person that you're sitting next to starts looking pretty attractive. You know? Well, you look at most mammals, they go into heat. The females are only ready to roll during the period of that estrogen spike and maximum fertility. Humans, it's a little bit different. We do have our ups and downs, but and they do center around the cycle, but we're not, human females are not completely shut off in between that, which is a good thing in mind. Okay. Okay. All right, so these are a few other points here. Now, the other thing about this is when you regulate multiple genes, you can have a single protein that can actually activate an entire developmental program. We mentioned an example, that so-called eyeless gene in fruit flies, or the equivalent of PAX6 gene in vertebrates. That gene, that single gene, when it gets turned on, there's a transcription factor that activates it highly. It's going to generate the formation of a complex structure, either the arthropod compound eye or the vertebra type of eye, which is a very different design. There is a specific transcription factor called MyoD that tells cells that would otherwise turn into the fibroblasts that make up connective tissue, stuff like that. It causes them to differentiate into muscle cells. So the reason you have muscles, you can thank the MyoD's family of transcription factors to do that. So, you know, that's a very complex developmental pathway, turning a generic cell into these highly specialized cells like the skeletal muscle cell. And it really depends on this particular small family of related specific transcription factors. They're found in the precursors of muscle cells, and that causes those cells to differentiate into muscle. So that's pretty cool. So you have that kind of thing with one single protein having a wide range of effects here. Okay, we have a few other general points about these guys. Okay, many of these transcription factors are not single proteins. Instead, they will bind to other transcription factors, sometimes close relatives, but sometimes quite different proteins. A given transcription factor may be able to bind to several other different transcription factors. And then this assembly, this complex, sticks out an enhancer and does something to transcription for that gene. And you can have very different effects depending on what you're bound to. If you're a transcription factor, I'll kind of draw it over here. And it binds to transcription factor A, whatever it is. Maybe that stimulates transcription. But if instead it binds to protein B, it represses it. So we can have these kinds of things, where depending, a given transcription factor, depending on what else it is bound to. Now some work on their own. Questions?
Okay, what is SPF?
Specific, oh, did I say SPF or SPF?
Yeah, it's here on number 5.
On number 5.
On number 5, it says SPF.
Oh, no, STF.
No, the
No,
first
the
one.
other one, the other
Oh, my mistake.
one. Okay.
Okay,
Yeah, I was trying to
specific
figure out which
transcription
one it was.
factor. Okay, sorry about that. Okay, all right. So we have examples. Sometimes a protein can bind to one protein, stick in from an enhancer, increase transcription, binds to a different protein, has exactly opposite effect. It's an inhibitor of transcription. We'll see some examples of that later on. Yes?
What's the M word?
The M word. Modulate. In other words, it can alter its effects in one way or another. In some cases also that will determine exactly what enhancers it will bind to. Now here again we see this in kind of a big thing with steroid hormones. Steroid hormones have different effects in different cell types. Using the estrogen thing as an example, we see that certain cell types are stimulated to proliferate. That's the ones in the uterine lining. Other cell types are stimulated to undergo, maturation, ovulation, things like that. And others are stimulated to alter, say, production of neurotransmitter receptors or something, basically strengthen or weaken neural pathways. Okay, same protein, different effects in different cell types. How come? Because those steroid hormones also bind to other transcription factors in various cells, and that determines what genes are going to regulate and whether they turn genes on or off. Most genes are regulated by steroids or turned on. In other words, transcription is stimulated. But in some cases, some of the genes might actually be repressed instead. So that's how you get the same molecule, the same estrogen affects many different cell types in different ways. And of course, that's one reason why people have to be careful about, high-dose steroids, whether you happen to be, what was his name, Lance Armstrong? Right. Aside from getting himself into serious academic trouble and losing serious political trouble and losing medals and stuff like that, it's also because high doses of testosterone and testosterone analogs are going to affect all kinds of different systems. He's putting his health at risk later on. One wonders, he's been doing it for a long time, whether his testicular cancer that he had survived, which is, he was very lucky, might not have been stimulated at least by all the stuff he was taking before. And he may have some nasty surprises down the way too if he keeps on doing it, aside from possibly being sent to the penitentiary. He could do that. He can go to jail for that. Okay, but at any rate. Okay, so that's an important kind of thing too. You're getting interactions between other things. Now, not only that, that adds additional complications. Not only that, another point here. Okay, spell it right this time. Okay, specific transcription factors that are in different enhancers. In other words, they don't have to be the same transcription factor or on the same enhancer. Here, we'll just draw it here. One over here, right? Okay. Okay, they can interact with each other if they're close enough together. That interaction could radically affect how these things are going to work. And here we're dealing with different proteins that are attached to different enhancers, right over here. So for instance, let's suppose that this one here tends to increase transcription. This one here on its own tends to repress transcription. But you put the two together, and maybe transcription will be increased a tremendous amount, far more than the effects of any of them on their own, or repressed really strongly. That can happen. When the proteins interact with each other, bind to each other, it's going to ultimately change the tertiary structure of those proteins. And what that does to its other functions, nobody can tell unless you do the experiment. So we do see this kind of thing where enhancers and the proteins bound to them can interact with each other. And then, it's okay if I erase these? Okay. And then, perhaps a last major point here. Specific transcription factors are going to be regulated proteins. In other words, they're not constantly in one state or doing one thing all the time. And once again, those of you who took cell with me, I emphasize protein regulation is critically important. And there's a wide range, just like any other kinds of proteins, there's a wide range of regulation. We already mentioned this one in some cases. Binding of other proteins, such as transcription factors and stuff, can regulate things. It may enhance transcription in one case, repress it in another. So that can be one kind of thing. Okay, binding of small molecules can regulate transcription factors. Think of the steroid hormones and the steroid receptors. The steroid receptors are inactive. They won't do a thing. You can put it right in a pile of DNA and a pile of enhancers, and on their own, they won't do jack. They'll just sit there staring at you. But add the appropriate steroid hormone, and now it will bind the appropriate enhancers and regulate the genes. Okay, so steroid hormones are perfectly good example. Some specific transcription factors change their location when they're activated. For instance, steroid receptors are a good example. Ordinarily, without the steroid hormone, those steroid receptor proteins are found just beneath the cell's outer membrane, or plasma membrane. Now, if you happen to be in the cytoplasm, and your job is to regulate genes, well, you're not going to be able to do much regulation if you're not anywhere close to the nucleus. But when the steroid binds, not only does it activate it as a transcription factor, it also changes it and gets carted into the nucleus where it will work. A common thing that BRCA1 and BRCA2 genes that are involved in certain types of hereditary breast cancer and things like that, one of the things that happens when you have mutations in those is its location is altered. Normally it stays in the cytoplasm, goes in the nucleus. In many of these mutations, it's stuck in the cytoplasm and can never go into the nucleus. which means the genes it regulates don't get properly regulated, and that leads to excessive growth. It's also found, I think, in ovarian cancers and stuff as well. It's not only restricted to one type of cancer. But part of the problem is the mutations, it doesn't get put to where it belongs. So you have that kind of. So changes in cellular location can regulate things. And then, of course, we have our old friends, our covalent modifications, things like phosphorylation. Many transcription factors are Okay, phosphorylation, covalent modification, attachment of phosphate groups to a protein. Many transcription factors have multiple phosphorylation sites. Multiple kinases, different kinases can phosphorylate or phosphatases remove phosphates from a given site, and each one of these changes is going to have an effect on how it interacts with DNA, whether it binds to an enhancer or not, and what it does once it is bound. And to a lesser extent, it's probably not all that common, but acetylation, the covalent attachment of negatively charged acetyl groups, will help to regulate some of them. But some of these transcription factors can be phosphorylated or dephosphorylated in multiple places on the same molecule. And what that does is that regulates it quite a bit. So these transcription factors are highly regulated proteins. And here again, you can have misregulation of a specific transcription factor, at least the wrong one, can lead to all kinds of problems. Developmental defects, implication in cancer, in certain cancers and things like that. So transcription factors are not something you want to mess around with. It's a general thing. Don't mess. This is a transcription factor. Dad, improper playing with this can be hazardous to your health or to your development. So that's a number of basic points, general points about these specific transcription factors. Now, we're going to see how they interact with the basal complex in just one moment.
Oh, boy.